One of the biggest challenges in medicine today is that it can take more than a decade and billions of dollars to develop a new drug. And most new drugs fail when they enter human clinical trials. A major reason for this is that drugs must initially be tested on animals, even though animals don't accurately replicate human biology. More often than not, results from animal studies are proven ineffective when the drugs enter human clinical trials. And to continue sacrificing animal lives in this way is extremely concerning. At the Wyss Institute, we set out to develop safer and more effective drugs, faster and at a lower cost, while also saving animal lives. To help accomplish this, we invented human organ chips, with our first breakthrough being a living, breathing lung chip. Organ chips are small micro-devices about the size of a computer memory stick. The chips contain tiny hollow channels lined with living human organ cells, interfaced with blood vessel cells, and they replicate complex functions of both healthy and diseased human organs. These devices mimic the flow of blood through vessels and the exchange of oxygen and nutrients between the blood and different tissues. They also allow the cells to experience normal physical cues, such as breathing motions in lungs. When the chips are lined by disease cells and drugs are pumped through the devices, the cells and tissues replicate responses seen in patients. Asthma, COPD, pulmonary edema, and cystic fibrosis, as well as the effects of cigarette smoke on human lungs, have been recreated in these chips. The U.S. government, foundations, and pharmaceutical companies immediately recognized the potential impact that organ chips could have on drug development. The FDA, NIH, and DARPA provided support to develop 10 different organ chips, with the goal of linking their blood vessel channels to create a human body on chip. The Wyss team beat this goal by developing a total of more than 20 organ chips, representing liver, kidney, intestine, heart, bone marrow, lymph node, vagina, and many others, and show that they can model numerous diseases, such as cancer and inflammatory bowel disease, as well as responses to drugs, radiation, toxins, and pathogens. The team also made personalized chips lined with different tissues from the same donor to explore patient-specific responses to drugs. Within 4 years of validating this work at the Wyss, Emulate spun out to produce organ chips. Emulate's organ chip systems are now being sold worldwide and used on-site at the FDA, as well as at more than 20 of the major pharmaceutical and biotechnology companies. Recently, Emulate demonstrated that their human liver chips are many times more effective at predicting drug-induced toxicities in humans than existing animal models. By replacing animal preclinical testing with human organ chips, the pharma industry has the potential to save billions of dollars and develop new drugs faster and more efficiently so patients can get the treatments they desperately need. By leveraging the Wyss translation model, human organ chip technology moved from a bio-inspired innovation to a commercial product, and it is now transforming research, drug development, and personalized medicine globally. Based in part on these advances, in December of 2022, Congress passed the FDA Modernization Act, which will now allow the FDA to consider organ chip data in place of animal testing when evaluating new drugs. In the coming years, we expect to see a wave of new, safer, and more effective FDA-approved drugs validated in human organ chips, which will save countless human and animal lives.